Welcome to our educational webinar titled The Importance of Maintaining and Assessing High-Quality Human Pluripotent Stem Cell Cultures. My name is Ashley Watson, and I'm a Product Marketing Manager at STEMCELL Technologies. I will be your moderator for this webinar. It is now my pleasure to introduce you to our speakers today. Dr. Adam Hirst completed his PhD in stem cell biology from the University of Sheffield, where his research focused on genomic instability in human pluripotent stem cells. Adam is currently a scientist at STEMCELL, developing products to facilitate innovative techniques such as gene editing in human pluripotent stem cells. Dr. Andrew Gaffney completed his PhD in pediatric oncology from the University of Leeds, UK, where he established a human embryonic stem cell disease model of Ewing sarcoma. At STEMCELL, Andrew monitors the requirements needed for establishing successful hPSC-based disease models, drug screens, and cell therapy applications. Let me now hand you over to Adam and Andrew to begin the presentation.
Okay, thank you, Ashley, and thank you for joining us for this live webinar today, where we're going to be highlighting the importance of maintaining and assessing high-quality human pluripotent stem cell cultures. So as an overview, we'll first be looking at the great potential of human pluripotent stem cells, also known as hPSCs, and move on to the implications that cell quality has had and is having on stem cell research. We'll describe some individual critical quality attributes of PSCs, and Adam will focus on one of these in particular, genomic integrity. Finally, we'll look at a rapidly developing modern-day application, genome editing, using the CRISPR-Cas9 system. We'll then hopefully have some time at the end for some questions. So the potential of human pluripotent stem cells is vast, and here you can see in this diagram that throughout normal human development, the inner cell mass of a 4 to 5 day old blastocyst can be isolated and placed into culture in vitro. Once established, these cells are known as human embryonic stem cells, or hESCs. These have the capacity for indefinite self-renewal and have the potential to differentiate into all somatic cell types derived from the embryonic germ layers, ectoderm, mesoderm, and endoderm. Alternatively, a somatic cell can be isolated from a patient and pluripotency factors added. Once expressed, these transgenes revert the somatic cell back to a state of pluripotency. With this method, the cells are called induced pluripotent stem cells, or iPS cells. Collectively, ES and iPS cells are called human pluripotent stem cells, or hPSCs, and both cell types are successfully being used to study basic principles of developmental biology, for regenerative medicine studies, as accurate models of human disease, and for tox screens and drug discovery. Now, PSCs are currently being used in a wide variety of clinical trials at phase 1 or 2A. And as you can see from this table that we compiled, which is accurate as of the end of 2017, the bulk of the trials are focused on developing a cellular therapy for age-related macular degeneration, the leading cause of vision loss in the over 50s. Trials are also ongoing for Parkinson's disease, spinal cord injury, and myocardial infarction. It's important to note that it's the differentiated derivatives of PSCs, such as retinal pigmented epithelium for AMD or cardiomyocytes for cardiac disease, that are being used in these trials, and not the undifferentiated PSCs themselves. Now, historically, a disorder would be categorized through epidemiological studies. Its inheritance pattern would be noted, which would allude to whether it has a genetic background. Genome-wide association studies could be performed to identify the specific genetic variants responsible for the disorder. And finally, sequencing has enabled us to elucidate individual mutations. Now, at the most, this gives us a good picture of the genetic status of a disorder, but we can't study the key biological mechanisms behind it without a good disease model. Fortunately, iPS cells can now be derived from patients which captured the genetic and epigenetic status of the starting cells, making them excellent disease models after being differentiated to the relevant cell types. Now, there are two types of cell therapy that iPS cells are being used for, and the first we're going to focus on is autologous cell therapy. This is a treatment method in which cells are taken from a patient's own body, reprogrammed back to a state of pluripotency, potentially manipulated by something like a gene correction, differentiated, and finally transplanted back into the patient. Now, because the cells originate from the patient, this may remove some of the risks attributed to immune rejection. Here you can see we've highlighted one clinical trial in particular that began in August 2013 and was headed up by Masayo Takahashi at the Riken Institute in Japan. And in this trial, they aimed to transplant a differentiated RPE sheet back into the retinas of patients suffering from neovascular, age-related macular degeneration. So in this study, iPS cells were generated from skin fibroblasts, obtained from 2 patients with an advanced form of AMD, and differentiated into RPE. Many QC checks were performed, such as analyzing DNA methylation and gene expression profiles, and immunodeficient mice were also used to test for any tumorigenic potential of the cells. Now, the trial ended up being very successful for patient 1, who saw no deterioration in vision 2 years after the surgery. However, the trial was actually halted for patient 2 after they identified 3 aberrations in DNA copy number that would probably affect the expression of genes encoded by the deleted DNA and the DNA flanking these regions. One of the 3 deletions was also on the X chromosome, and because the patient was male, it was decided that continuing the study would be risky. Now, despite the mutations, the cells did actually pass in vivo tumorigenicity testing, but they didn't proceed with the transplant due to a lack of understanding of the mutational effects. Okay, so the second type of cell therapy we're going to talk about is allogeneic therapy, and it's generally agreed that the process of taking somatic cells from every individual patient and reprogramming them is very labor-intensive, and this could be enhanced by creating healthy, ready-to-go iPS cell line banks constructed from donors. Obviously, we've now had a lot of experience from bone marrow transplants, and we found that matching 3 of the major HLA loci, and that's A, B, and DR, reduces the rate of immune rejection. Here you can see from the diagram on the left, taken from a paper by Shinya Yamanaka, that in theory, Japan requires about 50 unique HLA homozygous donors to cover about 73% of the whole population. And if you want to cover 90%, you need about 140 donors. So this is a much more realistic approach to applying iPS cell therapy en masse. And here's a story on the right of the first person to receive an allogeneic iPS cell therapy. And this was actually an extension of the previous trial I mentioned, where a Japanese male was treated for AMD. We're going to be given full details on how this patient did about 5 years after the transplant, so we'll be looking forward to these results somewhere in 2022. Now, the previous clinical trial showed just how important it is to analyze cell quality, especially before transplanting these cells into a patient. But it's also important to be cognizant of cell quality during all other areas of research as well. And here I want to highlight this paper from Nature Cell Biology in 2016, where they use this great quote, "iPS cell authentication and quality underlies their value at the frontier of biomedical advances." And what they mean by this is that we need to define what cell quality is and how to adhere to it before the great potential of these cells can be fully realized. They say that researchers need the assurances of strict quality control, conscientious tracking, and definitive identification to produce studies yielding reproducible data and valid results. The NIH has also recognized this as an issue, and after a workshop on reproducibility in cell culture studies, there are now grant guidelines that require authentication of cell lines before using them in any studies. It should also be noted that Nature journals have also announced cell line authentication policies as well. Finally, they state that we need to adopt the highest quality standards and verify and report iPSC properties. A key step towards this goal will be stem cell researchers reaching consensus on appropriate standards and best practices, and this is now being worked on by international consortia, such as the International Stem Cell Initiative, or ISCI, and the International Stem Cell Banking Initiative, also known as ISCBI or ISCBI. So what are the critical quality attributes of PSCs? Well, at Stem Cell Technologies, in line with multiple quality discussions and presentations from key consortia, we present 5 aspects of pluripotent stem cell health to be mindful of. These are morphology, gene and marker expression, the epigenetic landscape, pluripotency, and genomic integrity, and we're going to be going through a few of these with you in more detail today. Now, interestingly, it's actually quite difficult to find a consensus on the exact attributes of what a good quality PSC is, and that even applies to researchers working within the same lab. I'm sure we'd all love to be working with the best quality cells all the time, but we do have to be realistic about our limitations. This work takes time, it costs money, and the applications we use have to be sensitive enough to detect what we need to know. But above all, these cell quality attributes need to be considered by everyone working with PSCs, not just those researchers moving towards the clinic. Poor cell health can have a negative impact on any form of research, so we need to define this better and we need to enforce the message more. Now, firstly, as many of you will be aware, morphology is perhaps one of the most sensitive indicators of PSC quality. It's something that is often checked by researchers under the microscope daily. However, it is subjective and it's largely based on the experience level of the end user. Morphology can also differ depending on the specific culture conditions being used and also the pluripotency states that are being cultured. However, for cell lines that are being clump passaged, we're really looking for round, tightly packed cells with a high nucleus to cytoplasm ratio. The colony should have smooth edges and be circular, and there should also be some multi-layering in the center under a phase bright microscope. So to really test the sensitivity of this cell quality trait, we placed H9 cells into culture in standard MTESA 1 medium, as well as MTESA with a key factor removed. And as you can see from these images, the morphology started to really take a hit in the suboptimal medium. And this was reinforced by our morphology scoring assay, which we've developed in-house. And as you can see from this chart, the score started to decrease from passage 3 onwards. And this is largely based on the shape and size of the colonies, how spiky the edges are, if the cells are loosely packed, and whether there's any evidence of multi-layering. Now, interestingly, if we look at passage 5, TRON60, and OCT4 expression levels, you can see that they're actually very similar between MTESA 1 cells and those being maintained in the suboptimal medium. So this indicates that there's actually quite a large lag between morphology starting to score poorly to the downregulation of these PSC markers. So it's possible that morphology is actually the more sensitive trait. Now, even if this is true, it's important to note, as I mentioned earlier, that morphology can differ depending on the specific culture conditions being used. So here we've included some examples of H9 cells, and firstly here what they look like in a knockout serum replacement-based medium on inactivated mouse embryonic fibroblasts, or MEFs. And here you can see that the cells are very tightly packed together. Secondly, in MTESA 1 medium on matrigel matrix, where the cells have been clump passaged, and here you can see those nice morphology characteristics I was mentioning before. Next, in the same conditions, but where the cells have been single cell passaged, and as you can see here, they lose those traits and begin to form a monolayer. And finally, where they've been reverted to a naive-like state in reset medium back on inactivated MEFs, where they display a domed morphology rather than flat with refractive edges. Now, all of these morphologies are different, but it doesn't necessarily mean that one demonstrates a greater degree of cell quality over another. And because of this, it's going to be important to assess what morphology characteristics are meaningful when dealing with the many different culture systems that are available. The next cell quality characteristic is gene and marker expression, and we can split this up by the commonly expressed cell surface antigens, such as SSEA3 and 4, and the TRA markers, as well as developmentally regulated genes, such as OCT4 and NANOG. Now, it's true that undifferentiated cells should express these genes and markers. However, as I demonstrated on the previous slide, there's a question mark over how tightly these link to any variant in cell quality. RNA-seq isn't currently a routine measure due to the cost of processing samples. However, it can show some really useful data on differential expression between culture conditions, cell lines, and even individual cells. Similar cell lines from different studies can also be compared. And here you can see an example of some RNA-seq data where we've compared between 10 to 12 cell lines in multiple culture conditions, including a knockout serum replace-based medium on MEFs, as well as 2 feeder-free systems. In general, the expression patterns cluster within their relative culture systems, and as you can see from the orange cluster from the MEF system on the left, many endoderm and mesoderm specific markers are upregulated compared to that of the feeder-free media. So it's great to be able to gather such in-depth data on gene expression. However, even armed with all of this knowledge, it's very difficult to interpret whether a cell line is in fact good quality or not. Now, pluripotency has historically been assessed by the teratomer assay, and this is frequently considered as the gold standard method. In this assay, cells that are considered to be pluripotent are injected into immunocompromised mice, and their histology analysed for the presence of all three germ layers. Here's a nice image on the right from a paper from Ivan Damyanov and Peter Andrews, where you can see evidence of mesoderm from the cartilage and muscle, ectoderm from the neural tissue, and endoderm from the endodermal tubes. However, there is a lot of inconsistency in this method as every lab seems to perform it slightly differently. For example, the injection site, matrix, number of cells being used, the passage number of the cells, as well as a number of other attributes that all seem to vary. Many of the tumours can become cystic and are very hard to analyse. You'll require an experienced pathologist to analyse the samples, and the resulting sample frequently contains undifferentiated cells, potentially indicating teratocarcinoma. And here you can see an example of this, where the numerous cystic and tube-like structures are formed from undifferentiated cells which have condensed nuclei and scant cytoplasm. So without additional studies, these cells can't really be properly identified. So because of all of these factors, researchers are now seeking alternative methods to assess pluripotency, and one example is the quantification of PSC markers. And while it's true that OCT4, SOX2 and NANOG do correlate with the teratoma-forming potential of PSCs, it's important to know that these are markers of the undifferentiated state, not pluripotency. Again, I'll stress that an undifferentiated cell should express OCT4, however this doesn't show that the cell can differentiate to all three germ layers, so it's not indicative of pluripotency. Pluritest and scorecard are also frequently used. However, despite its name, pluritest can only measure the proportion of undifferentiated and differentiated cells in a mixture and can't inform you of the type of differentiation that's occurring, so it's not measuring pluripotency. For an informative readout, scorecard should be an endpoint analysis of a functional assay. Examples of these include in vitro differentiation methods of differentiation, and this technology is what our trilineage differentiation kit is based upon. So this kit is extremely simple to use and gives reproducible differentiation to all three germ layers across multiple PSC lines. It's a 1-week monolayer-based protocol, so no need to create EBs, and it's also amenable to high throughput. As the workflow at the top here shows, you simply create a single cell suspension and seed down the cells and empties of one medium at day 0. At day 1, you swap into ectoderm, mesoderm, and endoderm media, and then feed daily. Mesoderm and endoderm differentiation finishes at day 5, but ectoderm runs for a full week, and these additional 2 days are required for the PAX6 marker to be fully upregulated. So the chart on the left here demonstrates how reproducible this kit actually is, including data from 5 independent PSC lines. Ectoderm differentiation is measured by PAX6 expression, mesoderm by this T marker or brachyury, and endoderm by SOX17. As you can see, all markers have expression levels of over 80%, demonstrating that this kit is highly efficient. We also combine an additional marker for each lineage for ICC staining on the right, where we include nestin for ectoderm, NCAM for mesoderm, and FOXA2 for endoderm, and we're getting a really nice level of dual staining across the board. And here I wanted to include results of an experiment we performed in-house, comparing embryo body differentiation to our directed trilineage differentiation kit. So many researchers are validating pluripotency by allowing EBs to spontaneously differentiate in serum-containing media and assessing evidence of trilineage differentiation potential afterwards. Here you can see RNA-seq data of undifferentiated H9 cells, where red indicates high expression of markers, and green shows low expression. And as expected, PSC markers are being highly expressed in the undifferentiated cells, whereas lineage-specific markers are generally low. Now, interestingly, after EBs were allowed to spontaneously differentiate for 10 days, there was only a significant upregulation in ectodermal markers. So as a result of this particular experiment, we actually cannot say that this cell line is pluripotent, even though we all know that H9 cells actually are. After this, we ran the H9s through the trilineage differentiation kit, and you can see clearly that the directed nature of this assay with the nice tight gene expression patterns. So here the kit demonstrated pluripotency of H9s where the spontaneous EB assay did not. I'm now going to pass you over to Adam Hirst, who's going to take you through the subject of PSC genomic integrity and why this is such an important topic. So here you go. Thank you very much, Adam.
Thank you, Andrew. So I'm going to take you through a general overview of karyotypic instability in human pluripotent stem cells. So it was noticed that following prolonged culture in vitro, cells started to display decreased population doubling times as well as increased plating efficiencies. Now, these altered growth characteristics were linked to karyotypic changes observed using G banding. And as we started to find these, we found that they were presented in a non-random and sporadic manner. In subsequent studies, these karyotypically abnormal cells were shown to display a growth advantage over the normal diploid cells, often taking over the culture and becoming fixed within the population. It's important to note that these recurrent abnormalities are also observed in a number of human cancers. So our understanding of karyotypic instability come from two large-scale karyotypic studies published in 2011. So the first was by YCell, and what they did was they took over 1,700 samples and karyotyped them, and these were from 40 independent human embryonic stem cell lines and 219 independent iPSC lines. What they found was there was no notable differences between the incidence of karyotypic instability between human embryonic stem cells and iPSC lines, both of which present around 13% abnormal cultures. They also noticed no increase in incidence between cells grown in the presence or absence of feeder cells. The study did note that the rates of aneuploidy do increase with prolonged culture, so the longer you culture these cells, the more likely they are to become abnormal. However, there were some exceptions to the rule where they found normal cultures at very late passages, around 200 passages, and abnormal cultures at early passage, around passage 4. So this is sporadic. The paper also noted that trisomies 8 and 12 do predominate in human pluripotent stem cell culture. The second study by the International Stem Cell Initiative looked at 120 human embryonic stem cell lines which were karyotyped both at early and late passages. What they found was that 66% of these cell lines were normal. However, around 32% of these either gained an abnormality over time or were abnormal both in the early and late passages. In very few cases was an abnormality lost over time. Now, this is just showing that once a karyotypic abnormality is acquired by a cell line, it's very unlikely that it will be lost. The study highlighted that chromosomes 1, 12, 17, and 20 were recurrent in human pluripotent stem cell cultures. And again, that the rates of aneuploidy do increase at later passages, again with the same exceptions found with the YCell paper. The study also looked at small structural variants using SNP arrays, and these were to identify abnormalities which were present at lower magnification, so less than 5 megabases, which can't be detected by G-banding methods. What they identified was a duplication on the long arm of chromosome 20, which was identified in 22 karyotypically normal cell lines. So these cell lines were G-banded as normal. In 17 of the 22 instances, it was found only in the late passage, so this had been acquired over time in culture, and the remaining 5 it was found both in the early and late passage. Now, in subsequent studies, this region was found to confer a very strong selective advantage in hPSCs. Another paper I'd like to highlight is a paper from the Kevin Egan lab from 2017, where they looked at 140 banked human embryonic stem cell lines, 26 of which were prepared for clinical applications. They performed exome sequencing and identified 6 p53 mutations in 5 unrelated human embryonic stem cell lines. These missense mutations map to residues disrupted in a number of human cancers, highlighting the need for in-depth study of our pluripotent stem cell lines. This would not be detected by G-banding or SNP arrays. It's important to note that 95% of the cell lines were free of detectable p53 mutations. However, this is something that we should keep in mind. So this is an ideogram of the human karyotype showing all reported gains and losses from a paper by Barker et al. In 2016. Now this ideogram, the green bars represent gains of chromosomal material and the red bars indicate loss of chromosomal material. Now what's quite striking from this image is that you can see there are a lot more gains than losses, but also that these abnormalities present in a non-random manner. So you get frequent gains of chromosomes 1, 12, 17, and 20, the abnormalities that were identified by the International Stem Cell Initiative. So fixed karyotypic abnormalities in our cultures rely on 2 distinct factors. So first of all, the cells have to acquire a mutation. The mechanisms behind these mutations or chromosomal abnormalities are relatively unknown, and the mutation rate is very hard to study, so we don't know what it is in our PSC cultures. Once the cell has acquired a mutation, it then has to create a growth advantage over the diploid cells, so selection has to take place. Now this is something that can be measured using mixing assays, so using fluorescently labeled abnormal cell lines, we can actually track these cells over time and see how they're taking over. If we look at the types of aneuploidy that we do observe in PSC cultures, we can start to elucidate potential mechanisms underlying karyotypic instability. So the most abundant abnormality that's observed are numerical aneuploidy. So this is the gain or loss of individual chromosomes. And this suggests errors in mitosis, so chromosomal missegregation during anaphase. So this typically results in monosomies or trisomies. We also see structural aneuploidies within our PSC cultures. These are unbalanced structural rearrangements or translocations between chromosomes. And this suggests non-homologous recombination events between chromosomes or errors in DNA repair mechanisms. We also observe submicroscopic changes. So these are copy number variants and point mutations, which these are unlikely to be detected by G-banding. However, these suggest errors in DNA synthesis and or repair. So if we look at what a human pluripotent stem cell can do in culture, it can self-renew, they can differentiate, and they can die. So if we look at how different genetic variants are selected for, we can start to elucidate what attributes we're actually selecting for. So we're going to be selecting for cells which have reduced cell cycle time, so cells that divide faster. We're also selecting for cells which have increased cell attachment. So we're constantly passaging these cells, so we're selecting for cells which stick down into the new vessel faster. We're also selecting for cells which have reduced differentiation, so the cells that do differentiate in our cultures, we typically remove them and don't carry them on to the next passage. And we're also selecting against cells which have a high propensity to die. So cells that tend to avoid these cell death mechanisms, like apoptosis, in the case of the 20q abnormality, these cells rapidly take over the culture. Not only is our hPSC culture system very selective, we also have a very selective workflow. So if you can imagine you're starting with a somatic cell in PSC culture, you could potentially have mutations within these somatic cells, which are donor dependent. You then reprogram these cells back to an induced pluripotent stem cell state. And in this method, it's a very low efficiency, so you're talking less than 0.1% efficiency. So you're selecting for a subset of cells that can actually become iPS cell lines. Many labs are still using integrating vectors, so this can cause insertional mutagenesis within the cell lines. And inherently, we always choose our best growing cell line, or the cell line that looks the best. Now, what are the attributes behind why this is the best cell line, and is this the best way of doing it? Once you have derived your cell lines, or you have an embryonic stem cell line, you undergo extensive passaging. So depending on what type of study you're doing, this could be quite a long process. You also might undergo a number of freeze-thaw cycles which introduce selection. A lot of people are now doing genome editing with these cells. So in this system as well, we're putting the cells under a lot of pressure. So as we transfect the cells, this is a very stressful process. Not many cells survive this process. We go through rounds of single cell plating. There are potential off targets using gene editing methods. We also have to go through clonal derivation to derive cell lines which have a specific mutation, and then we have to expand these clones again to work with them. The last part, which potentially could be a selection process, is differentiation. So we do observe a lot of cell death when we differentiate to particular lineages. So are we selecting for cells which have the ability to differentiate down that lineage? Another important point to bring up is a poster from YCell, which presented at ISSCR in 2017. And they looked at over 7,000 hPSC karyotypes. And in these karyotypes, they found over 1,300 recurrent abnormalities in over 1,000 karyotypes analyzed. Now, if we look at the trends of these over time, so we're looking from 2009 up to 2016, we do see an increase in chromosome 1q gains, an increase in 20q gains, and a decrease in 12p gains. Now, this could potentially suggest that changes in methods of derivation of cell lines or culture conditions or passaging techniques could be influencing the genetic landscape of our cells. This also may be the case of increased number of cell lines being analyzed due to the amount of iPS lines being derived from a number of donors. So how do we assess the genomic integrity of our cell lines? So the gold standard is G-banding, and this is what a lot of people use to karyotype their cells. Now, this detects a lot of the aneuploidies that we do see within our cultures that we observe. However, it doesn't detect submicroscopic abnormalities, particularly with the case of the 20q abnormality. The detection limit cannot go below around 15% of cells, and it takes a while, often involving sending the samples off to an external site. There are other methods which can be used alongside G-banding to catch abnormalities that we can't normally detect, and these include microarray to look at using SNPs or array CGH, and these can detect these CMVs, particularly with the 20q that we identified earlier. You can use fluorescence in situ hybridization to look at very specific sequences of interest, or people are now talking about using sequencing to analyze our cell lines, which will pick up a lot of the aneuploidies that we do observe. However, these can all be cost prohibitive, and it takes a long time to get results back. So the question that we get asked a lot is, how frequently should we monitor our genomic integrity of our cells? Well, you can imagine that if you have a cell line that you're starting your experiments with, which has been karyotyped and is normal, you may go through an extensive passaging period between the time when you receive the cells to when you analyze them at the end. And if you do find an abnormality, there's no way of checking when this abnormality arose throughout culture, potentially ruining experiments which have taken place between this point. So to address this problem, we developed the hPSC genetic analysis kit, which can be used to frequently monitor the genomic integrity of PSC cultures. So this is a qPCR-based method to detect recurrent abnormalities identified in the studies previously. It does not require sending samples away for analysis, so anyone with access to a qPCR machine can assess their cells. And with the kit, you're able to analyze around 20 samples at $28 per sample. The workflow of the genetic analysis kit is very simple. So once you've harvested your genomic DNA, you measure and adjust the concentration, prepare 2 master mixers, transfer this into a qPCR plate, and run the qPCR. And the excellent thing about this kit is that you can take genomic DNA from a cell line, extract it, and analyze the results the same day. So these are the regions that we picked to include within our kit. So we've included chromosome 4 as a control region, which is the least affected chromosome in the genome. We've also included 8 genetic assays designed to these minimal critical regions on the recurrent chromosomes identified. And we estimate this will cover around 75% of all reported abnormalities. We've also validated this kit using cell lines with known abnormalities. So you can see here, this has the cell line was identified with a chromosome a long chromosome 1 long Q gain. So this is a translocation between chromosome 1 and chromosome 21, where the long arm of chromosome 1 has translocated. The second one shows a 10p deletion, and this was identified again by G-banding. You can see the deletion of the short arm on chromosome 10. We can also detect trisomy 12 within a cell line. And the most important one that we're able to detect with the qPCR machine is this amplification of 20q1121. Now, in this case, the G-banding came back normal. So this would have been reported as normal if we'd have only used G-banding. However, we picked this up in using our qPCR kit, and sending the same sample off FISH analysis, you can see the duplication was detected in around 150 out of 200 cells. So this is really highlighting the importance of going deeper into the cells other than just using G-banding. Now, what we find with this 20q1121 mutation is it is cropping up a lot within our cultures. And what we've seen is this duplication being identified in a number of PSC cultures. And these are predominantly in a lot of single cell passage cultures. When we send these samples off after we've identified it, a lot of the time this karyotype comes back as normal. However, when we FISH the sample, you can see in this one, we were able to detect around 36% of 20q1121 within a culture. The kit also allows frequent screening, so if required, it could be analyzed every passage. However, you can see this is chromosome 4 control, which doesn't change over time in passage. However, we started to detect 20q abnormality around 21 passages, becoming statistically significant later on. This is a chromosome X, so you can see this is stable again over time. This is just an example of one of the other chromosomes. And you can see this is a male line, so we can also detect between male and female. The hPSC genetic analysis kit also detects around 30% mosaicism. We're also doing some further studies to see how low this is. However, you can see we mixed known percentages of abnormal lines into a normal culture and analyzed this by flow cytometry to get the exact number of abnormal cells. And you can see we're able to detect significantly around 34%. Around 26% is approaching significance, and further study will be needed to correctly identify exactly the mosaicism we can detect. So to summarize, karyotypic instability does affect growth, differentiation, and cell survival, particularly within our PSC culture conditions. Little is known about the downstream effects of these karyotypic instability. So if you have a cell line which has an abnormality which is undetected in the pluripotent stem cell state, once you differentiate that to a particular lineage, there's no evidence to suggest whether or not that cell line will be clinically applicable. Early detection is critical, so this qPCR-based method can be done every passage if desired. Now, what should you do if you detect a karyotypic abnormality within your cultures? So important cell lines can be rescued if there are normal cells present. So this early detection is critical. So you can subclone the cell line if you have remaining normal cells. There are elements of risks with that technique because you are subculturing and deriving cell lines from single cells. It is important to keep master and working cell banks and to go back to earlier thaws frequently. And what we want to do is really increase awareness of karyotypic instability in all hPSC labs, as we feel this is critical to understand the impact of genomic instability on all hPSC research, not just those people that are going towards clinical trials. So I'm going to hand you back over to Andrew, who's going to go through the genome engineering side of this presentation.
Okay. So returning back to methods of studying disease, I mentioned earlier that iPS cells can be isolated from patients and differentiated to create accurate models of human disease or to generate cells to be used in drug screens. Alternatively, normal PSCs can be formed from healthy donors or by placing human embryonic stem cells into culture. Genome editing tools such as ZFNs, TALENs, and more recently the CRISPR-Cas9 technology are now frequently being combined with PSCs to either correct or introduce disease-specific mutations. Once again, these cells can be differentiated downstream to create a disease model, produce cells for drug screens, or to create corrective cells for cellular therapy. Now, there are two additional cell quality characteristics that need to be considered upon the genome editing of PSCs, and the first comes as a result of PSCs demonstrating very low single-cell cloning efficiencies. More recently, novel applications such as genome editing require the generation of clonal PSC lines. However, a number of bottlenecks have been identified which restrict single-cell survival. And the first is simply the number of cells that survive post-plating. Following this, the surviving cells need to re-enter the cell cycle, and they do so at a relatively low rate. Finally, the cells that have successfully done this must survive past the first division, and by the time all of these bottlenecks have been applied, we end up with a cloning efficiency of less than 1% for the majority of PSC lines. Now, to help remedy this problem, we've developed Cloner, a supplement for teaser media which enables the robust generation of PSC lines. There's no adaptation to single-cell passaging required, so we won't be selecting for karyotypically unstable PSCs. It's compatible with the complete teaser family, as well as your choice of cell culture matrix. And finally, Cloner has now made the gold standard of low-density seeding possible, and this is known as single-cell deposition. The Cloner workflow is tremendously simple and involves PSCs being seeded as single cells at time 0 into a teaser media supplemented with Cloner. The cells are fed with teaser and Cloner at day 2, and by day 4 we return to feeding with your standard teaser maintenance media without Cloner. Colonies will be ready to pick at approximately day 10 to 14, which is a much earlier time point than by using teaser media alone, with or without supplementation with ROCK inhibitor. So this image really gets across the impact that Cloner has on clone establishment. And as you can see, 15,000 cells were seeded into 10 centimeter dishes in mTeaser1, supplemented with either ROCK inhibitor on the left or Cloner on the right. The number of clones was increased from 95 up to 2,398, a significant difference. However, as you'll see from the plates on the right, many colonies have actually merged into each other, indicating that mosaics have formed. Furthermore, if this occurs in the plate supplemented with Cloner, it's pretty much guaranteed that the colonies established with ROCK inhibitor are also mosaics. So this cell density is not a good one for either condition. We then dramatically reduce the seeding density to just 300 cells per plate. That's just 5.5 cells per centimeter squared. And you can see that we derived over 80 clones when supplementing with Cloner, but just 3 clones when using ROCK inhibitor. This is a true clonal density, and it's very unlikely that any of these colonies have developed as mosaics. Here's the data to back those images up, and you can see that for the 2 methods of generating clones, be it low density seeding or single cell deposition, we've managed to increase the cloning efficiency from less than 5% with ROCK inhibitor to 26% and 21% respectively. As a result, you can be much more confident in the validity of the clones that you're generating, and you can also perform the whole cloning procedure in a much more efficient manner. Now, we've recently launched a range of products for CRISPR-Cas9 genome editing of pluripotent stem cells, and this is known as the Architect CRISPR-Cas9 system. We've gone on to develop a standardized protocol which includes these reagents as well as the Cloner supplement. So to begin with, CRISPR RNA is designed and annealed to tracer RNA. The ribonucleoprotein or RNP complex is then formed by combining this with Cas9. The complex is then delivered into PSCs using either chemical transfection or electroporation. We've actually detailed the steps for both methods in our protocol, and the cells are plated initially at a high density in teaser media containing Cloner. The cells are then left for about 48 to 72 hours to allow the editing to occur, and they're plated at a low density in order to derive clonal lines. These clones are then established and characterized, and it's important to know that the earlier cell quality characteristics such as morphology, genome marker expression, pluripotency, and genomic integrity must once again be assessed prior to experimenting on genetically modified cell lines. You have to know that what you're working with is indeed a healthy PSC line. Now, if you'd like to read the in-depth protocol, you can find this under the Scientific Resources section on any of the Architect product pages on the Stem Cell Technologies website. Finally, I wanted to touch on one final aspect of PSC quality during genome editing, and this involves the potential for off-target effects of CRISPR-Cas9. Now, in its most minimal form, the CRISPR system functions with three components, and that's Cas9 endonuclease, a specificity-determined CRISPR RNA, and a transactivating RNA, also known as tracer RNA. The CRISPR and tracer RNA anneal to form the guide RNA, and this 20 base pair sequence, which has been designed to be complementary to the DNA target, is followed by the protospacer adjacent motif, or PAM site, typically taking the form of NGG. Now, although the targeting specificity should be tightly controlled by a 20 base pair guide RNA sequence, as well as a PAM site adjacent to the target sequence in the genome, potential off-target cleavage activity could still occur on DNA sequences with 3 to 5 base pair mismatches. Furthermore, studies have shown that different guide RNA structures can affect the cleavage of on-target and off-target sites. Other studies have demonstrated that while the PAM site is required for initiating the binding of Cas9, the seed sequence directly adjacent to the PAM site is critical for the activation of nuclease activities in Cas9. Now, there's a number of methods to identify the potential off-target effects, and one is in silico analysis of the guide RNA design. And this cell stem cell paper by Hendricks et al. In 2016 lists a number of guide RNA design websites which can include off-target prediction models. However, in silico methods do not fully predict all off-target sites, and so alternative methods of detection can be used ranging from simple assays with poor detection, such as the T7E1 assay, to more in-depth techniques such as deep sequencing. Finally, there's a number of strategies that researchers are now employing to reduce the number of off-target effects, and these include modifying the guide RNA design and finding a balance between on-target and off-target activity, controlling the concentration of the Cas9 guide RNA complex by titrating what's being delivered, and using an alternate form of Cas9 known as D10 mutant nickase, in which single-strand breaks are introduced. And as a result of this, twice the amount of Cas9, as well as 2 separate guide RNAs, need to be introduced into the system in order for it to be successful. There's also a number of other strategies, as well as new and improved methods, such as the use of high-fidelity Cas9 nuclease, that are continuing to be developed and are outside of the scope of this presentation today. However, with the rapidly expanding field of genome editing demonstrating just how powerful it can actually be, especially when combined with pluripotent stem cell research, we'll be watching the developments very closely to see the recommendations for how best to move forwards. So to summarize, the cell quality attributes we described, such as morphology, pluripotency, genome marker expression, and genomic integrity, should be monitored frequently in order to ensure the highest quality of cell cultures being used. It's critical to assess the genomic integrity of PSCs. This is because the process of routine PSC maintenance is highly selective, and genetically variant cells may affect rates of cell growth, differentiation potential, and cell death. It's important to note that there's no one single way of detecting all karyotypic abnormalities within PSCs, so the most efficient techniques for your research purposes should be identified. And finally, CRISPR-Cas9 genome editing has become a very powerful tool for researchers working with PSCs. There are 2 additional cell quality attributes that need to be considered in this case, including the derivation of clonal lines to ensure they're not mosaics, and the off-target effects that must be carefully considered and assessed.
Thank you very much, Adam and Andrew. That concludes the presentation segment of the webinar. We will now begin our Q&A session by addressing the written questions that were submitted throughout the presentation. let's start with our first question. So in the hPSC study, what was considered to be early and late passage?
So in the International Stem Cell Initiative study, the researchers asked for early and late passage cell lines, paired cell lines, and that wasn't defined what was early and what was late. The reason why they asked for early and late was to see if cells acquired a change over time in culture. So the exact passage numbers are not defined, but we are looking at one population of cells over a number of passages and then looking at that later passage.
Okay. Next, does the human pluripotent stem cell genetic analysis kit include positive and negative control samples?
The hPSC genetic analysis kit is provided with a negative control sample. So this is used to normalize sample genomic DNA to that control sample. So we don't include positive controls as well. It just comes with the negative control.
Okay, next question. What is considered best practice for monitoring the genomic integrity of pluripotent stem cell lines? Would you say it's every 5 passages, every 10?
So again, this is another question that's quite tricky to answer. Again, if we're talking a number of passages, this could be, people could be passaging twice a week, others could be passaging once every week. So I think the real answer to this question is as frequently as possible. You can see if you karyotype a cell line at the beginning of a study and then at the end of the study and you find an abnormality, you don't know when during that long period of time when this abnormality arose. So if we're able to monitor them a lot more frequently, we might be able to get a handle and stop these changes happening sooner if we can identify them earlier.
Okay, next, are there any functional tests that can be used to test the quality of PSCs?
So I think it depends on what your interpretation of a functional test actually is. So we can create embryonic bodies, which I'd say is one of the least stringent ways of assessing pluripotency. Then the next level up would be the teratoma assay. But both of these are limited because they don't really detect whether or not the cells allow for normal development in vivo. And that's why these are just called pluripotent stem cells, because for ethical reasons, they can't be tested in primates to demonstrate the normal tissue development. So in the absence of that, as I showed before, we offer the trilineage differentiation kit, which is a good alternative to demonstrate that these cells can differentiate to all 3 germ layers without needing to use animals. But I'm not sure whether you can strictly say that that's technically a functional test.
Okay, the next question is, how efficient is the NEON system for electroporating pluripotent stem cell lines?
So I think this brings up an interesting topic on what people are referring to as the efficiency during genome editing. And there's really 3 efficiencies that are being talked about and maybe a bit confused. So the first one is the transfection efficiency, which I think this question is referring to. And this is the number of cells that are actually being successfully transfected with the CRISPR construct. And with the NEON system, we're routinely getting over 90% efficiency in-house, and that's assessing GFP-positive cells by FACS. We've actually made a protocol for this in the technical bulletin I mentioned earlier, as well as for chemical transfection as well. So you should check that out, see what variables we used. The second would be the cloning efficiency, and that's the number of single cells that are successfully sitting down on the bottom of the plate, eventually growing into colonies. And it's the cloning efficiency that's improved using the cloner reagent that we discussed earlier. So with this, we're improving the efficiency from about 1% to about 30 to 40%. And then finally, there's the editing efficiency as well, and that's the number of cells that have successfully been cut and edited using the CRISPR system. And with the Architect product line, we're routinely getting about 30% cutting efficiency in PSCs.
Okay, the next question is, what are your recommendations for QCing genome edited lines?
Okay, so let's say you're starting off with an arbitrary number of cell lines, like or clones, like 100 clones, for example, first you need to screen out the ones that haven't been edited using an appropriate method, such as the T7 assay. You could also sequence your region of interest to remove any clones that are formed as mosaics. And then from the clones that are remaining, you might want to whittle it down to, say, your top 5 clones. And to do that, you'll need to check the quality characteristics that we went through in this presentation, like morphology and gene and marker expression. And you can use the hPSC genetic analysis kit to screen for the most common genetic abnormalities. And at the same time, it would also be worth checking for any off-target effects as well by sequencing something like the top 5 most likely regions. And I'd say that with all of that, you've really done your due diligence to end up with the best quality gene-edited lines to take forward into your experiments.
Okay, great. That concludes the Q&A session of the webinar. I'd really like to thank everyone for attending, and I'd especially like to thank you both, Adam and Andrew, for your time and a really excellent and informative presentation. Thanks again, and goodbye.